Hello everyone and welcome to the episode 3 of this mini series on mutations on my channel Micro and Mall Bio with Dr. Sunita Bundale. We have discussed substitution mutations in the first episode and frame shift mutations in the second episode. In today's lecture, we will be discussing suppressor mutations. If you like the content, subscribe and keep watching and keep learning. Suppressor mutation is a mutation that suppresses the phenotype of another mutation. For example, the first mutation occurs in the wild type and converts it into the mutant. Now, another mutation occurs in the mutant such that the effect of the first mutation is cancelled or nullified, leading back to the original phenotype. This second mutation is called the suppressor mutation. The exact nucleotide sequence of the wild type DNA may not be restored, leading to a changed genotype, but the phenotype is restored. These strains harboring suppressor mutations are phenotypically indistinguishable from the wild type strains. The most common type of suppressor mutation is reversion. Here, the second mutation in the mutant converts it back to the wild type with the same genotype and same phenotype. Bacterial strains that contain reverse mutations are called revertants. Let us move ahead and talk about the types of suppressor mutations. Suppressor mutations can be intragenic or intergenic. Intragenic suppressors are located in the same gene as the forward mutations that they suppress. The possible locations and nature of intragenic suppressors are determined by the original forward mutation and by the relationships between the primary structure of the gene product and its biological activity. Intragenic suppressors can be further divided into intracodon intragenic suppressor mutations and intercodon intragenic suppressor mutations. Let us find out about intracodon intragenic suppressor mutations. Intracodon intragenic suppressor mutations are such that the first mutation and the second suppressor mutation both occur in the same codon of the gene. The position of the mutations within the codon may be different. For example, in this figure, the third amino acid in the protein is leucine. A point mutation in the third position of 5' AAT 3' changed UUA to UUU, leading to a missense mutation and incorporation of phenylalanine in the place of leucine. If a second mutation occurs in the same gene but at a different position, here first position, then triple U changes to CUU, which codes for leucine. Thus, the original amino acid is restored in the protein and thus the original phenotype is restored. However, here the original genotype is not restored. The suppressor mutation has, however, nullified the effect of the first mutation, and both mutations have taken place in the same codon of the gene, and hence it is called intracodon intragenic suppressor mutation. So then what are intercodon intragenic suppressor mutations? Intercodon intragenic suppressor mutations are such that the first mutation and the second suppressor mutation both occur in the same gene but different codons. For example, in this figure, a protein has a negatively charged amino acid aspartate and a positively charged amino acid lysine at a distance. The protein structure is stabilized due to the interaction between aspartate, which is a negatively charged amino acid, and lysine, which is a positively charged amino acid. Now, a first mutation has occurred and changed aspartate to histidine, a positively charged amino acid. As a result, the protein loses its structure and probably becomes non-functional. A second mutation now takes place such that lysine is converted to glutamic acid, a negatively charged amino acid. This glutamic acid can interact with histidine in the same way and restore the protein structure as well as its function. The second mutation is a suppressor mutation, and because it occurs in a different codon of the same gene, it is called intercodon intragenic suppressor mutations. We will now move ahead and talk about the intragenic frameshift suppressors. We have already studied in episode 2 of this mini-series that a frameshift mutation occurs due to insertions or deletions resulting in shifting of the reading frame. An intragenic suppressor is a second mutation in the same gene that cancels the effects of the first. In this example, an insertion mutation leads to a frameshift mutation. A second deletion mutation resets the reading frame back to the original. Although there is an alteration of two codons, there is no frameshift. Let us now try and understand what intergenic suppressor mutations are. Intergenic suppressor mutations are such that they occur in gene different from the one in which the first mutation took place and whose effects they suppress. Intergenic suppressions are best studied in nonsense mutations. Let us see this example to first understand the events leading to a nonsense mutation. A first substitution mutation takes place whereby T in the gene is substituted by A. As a result, the sense codon AAG for lysine is converted to UAG, a nonsense codon. This results in a nonsense mutation and the process of translation stops and a truncated protein is formed. During translation, the decoding is brought about by tRNA, which interacts with the codon and brings the appropriate amino acid. In the figure, in the left panel, lysyl tRNA with CUU anticodon reads the codon AAG for lysine and incorporates lysine in the growing polypeptide chain. Now, because the sense codon AAG is changed to UAG, the lysyl tRNA cannot read it as its anticodon. Further, UAG is a stop codon and release factors bind to it and stop the translation process. Now, this nonsense mutation can be suppressed by another mutation in the tRNA gene. The anticodon of tyrosyl tRNA is GUA. If a mutation occurs in the tRNA gene for tyrosin such that the anticodon changes to CUA, then this tRNA cannot recognize the codon UAC for tyrosin. But this changed anticodon can now bind to stop codon and incorporate a tyrosine, and the polypeptide will continue to be synthesized as shown in the figure. Rather than a truncated protein, a protein with tyrosine in the place of lysine is synthesized. This small change may or may not have an effect on the function of the protein. Thus, a mutation in one gene is suppressed by a mutation in another gene, that is the tRNA gene. The suppressor mutation can be in any tRNA gene where a single substitution mutation changes the anticodon such that it starts recognizing a stop codon as its codon. We have seen that the substitution mutation can convert a sense codon to a stop codon, leading to a nonsense mutation. This nonsense mutation can be suppressed by a mutation in the tyrosyl tRNA, which can bind to UAG and suppress nonsense mutation. In a cell with a suppressor for UAG, say a UAG stop codon is present at the end of the mRNA, which is the genuine stop codon of that mRNA. Is it possible that the suppressor DNA will also bind to this then? And if this happens, then translation will continue and read-through proteins will be synthesized. Indeed, such read-through proteins are found in nonsense suppressor cells, but the cell has a way of protecting this from happening. Usually, there are two termination codons in a row on the mRNA where the translation is supposed to be terminated. Thus, UAG and UAA may occur one after another as shown in the figure. Even if there is a UAG suppressor, UAA will bring about termination of translation in this particular cell. In this example, suppressor mutation was in the tyrosyl tRNA such that it cannot recognize the UAC codon for tyrosin but can read the stop codon UAG. This means that if there are other UAC codons in the mRNA, they would not be read by the mutated tRNA. In such a case also, the translation may terminate. But this too does not happen in this cell. This is because tRNA genes are redundant. Mutation in one tRNA gene for tyrosin may give rise to an altered mutated tRNA which will recognize the UAG stop codon as its sense codon, but other tRNA genes which are normal will produce tRNA which will correctly read UAC as the codon for tyrosin and translation will continue. After having discussed nonsense suppressors, let us now discuss missense suppressors. We have discussed missense mutations in episode 1, where we have seen that these mutations alter a codon in a manner that a different amino acid is incorporated in place of the original one. The figure on your screen shows how GGA codon is read by glycyl tRNA with its anticodon UCC, and glycine is incorporated in the growing polypeptide chain. Now a mutation converts the GGA to AGA, a codon for arginine. As a result, arginyl tRNA with its anticodon UCU reads the codon AGA and incorporates arginine in the growing polypeptide chain instead of glycine. A missense mutation has taken place. This missense mutation, however, can be suppressed by intergenic suppression in more than one ways. The first mechanism is similar to the nonsense suppression that we have just seen, where a corresponding mutation in the anticodon of the tRNA takes place. Thus, a mutation in glycyl tRNA anticodon may convert it from UCC to UCU such that now it recognizes AGA as its codon and incorporates glycine. Thus, the missense mutation is suppressed. However, a second suppressor mechanism may also occur in amino-acyl tRNA synthetase gene. Amino-acyl tRNA synthetase is an enzyme which is responsible for the loading of the amino acid on the tRNA. It has to correctly recognize its cognate amino acid and its cognate tRNA. For example, phenylalanine tRNA synthetase has to recognize its cognate amino acid phenylalanine and phenylalanyl tRNA. It will correctly load phenylalanine to the acceptor arm of the phenylalanyl tRNA. During translation, this tRNA recognizes the codon UUU on the mRNA and phenylalanine is added to the growing polypeptide chain. At the late stage of translation, only codon-anticodon interaction matters. The translation machinery does not check which amino acid is loaded onto the tRNA. Thus, missense mutations can be suppressed where a mutation in tRNA synthetase occurs such that it correctly recognizes its cognate tRNA but charges the tRNA with the wrong amino acid. Let us understand this with our example where a missense mutation has converted GGA, which codes for glycine, to AGA, which codes for arginine. This is arginyl tRNA with anticodon UCU for arginine. A mutation in arginyl tRNA synthetase occurs such that it correctly recognizes its cognate tRNA but charges the tRNA with glycine instead of arginine. This arginyl tRNA loaded with glycine will read the AGA codon and incorporate glycine, suppressing the missense mutation. This arginyl tRNA can also have a mutation in glycyl tRNA synthetase such that it recognizes arginyl tRNA as its cognate tRNA mistakenly and charges it with glycine. This too will bring about suppression of the missense mutation in the same way because it can read AGA as its codon. Let us now move on and talk about intergenic frameshift suppressor mutations. We have seen in the previous lecture how insertions and deletions can cause frameshift mutations. This is an mRNA sequence. In this mRNA sequence, addition of a base G will shift the frame, leading to a frameshift mutation. Intergenic frameshift suppressor mutations depend on a suppressor tRNA which can restore the reading frame. For this, it should have 4 bases in its anticodon loop. One such example is that of glycyl tRNA. Normally, this tRNA has CCC as its anticodon, but the suppressor glycyl tRNA has 4 Cs in its anticodon loop. Thus, if in the mRNA sequence a G has been inserted after GGG, then this 4 times G can be read by this suppressor tRNA with its CCCC anticodon as a codon of glycine. This results in the suppression of frameshift mutations. With this, we come to an end of this lecture. We have discussed suppressor mutations today, where we have dealt with intragenic and intergenic suppressor mutations. In the previous episodes of the 3-episode mini-series, we have discussed point mutations and frameshift mutations. If you like the content of this channel, like, share, and subscribe. Thank you.